Let's make it happen. There's still work to be done, right? And everybody in this room can make it happen. So let's work together, and that is our guiding report. All right, so I want to introduce our next panel. It's the Science and Research in Biscayne Bay, an update from the field, moderated by Dr. Natalia Soares Quinete. Welcome, Natalia.
Listo. I have a clicker for you here. Sure. Clicker. Okay.
For the yep.
Let's sounds good.
So, good morning, everyone. I'm Dr. Natalia Soares Quinete. I know last name is hard to pronounce. I'm an associate professor in the FIU Institute of Environment and also part of the Department of Chemistry. And my research also focuses a lot in Biscayne Bay, closely to the environmental chemistry and trying to address emerging contaminants. I was actually here the last time, also was one of the speakers, so I'm very excited to be the one moderating this time. I work with PFAS. I think probably most of you know as forever chemicals. So a lot of the research that I do goes on in all the South Florida ecosystems, aquatic environment, including the most important part of this summit here, that's Biscayne Bay. And over the past several years, my group has been actually working on understanding it, PFAS occurrence, distribution, transport, and also ecological exposure impacts in the bay and also in the tributaries. We actually been some of the first ones to determine PFAS in surface water from the bay, groundwater, and also atmospheric deposition. And actually, we're going to have a talk from one of my graduate students, past graduate student. She's already graduated. So I hope you guys are going to be able to attend it in the afternoon. So again, not delaying a little bit, just as you all know, Biscayne Bay is, a complex, dynamic system, and one that requires, integrated monitoring it to understand how water quality, nutrients input, seagrass conditions, and also ecological community responsible chronic pressures as well as acute disturbance. Today we have the science and research panel bringing together a group of scientists who are deeply involved in the monitoring of the bay and trying to understand how these ecosystems are changing in real time. I'm actually very excited to be here and hear more about and highlighted all the scientific work that is being done in the bay. And without delaying further, I want to introduce the panelists. We're gonna have here Dr. Kassidy Troxell, Dr. Rolando Santos, Catherine Macdonald, Griffin Alexander, and Laura Eldredge. And I'm going to be introducing one by one. You guys want to come in here? Just come in.
Yes, dear.
Please.
Jump.
So our first speaker is going to be Dr. Kassidy Troxell. She's a research assistant professor in the Institute of Environment at FIU, and she's going to begin talking more about nutrients and contaminants and microplastics in the bay. Her work provides some essential context in understanding how nutrients and contaminant dynamics influence ecosystem health and environmental quality. And I guess I have to change here, right? Oh, no, this is just a...
How do we change that?
Oh, there it is. Oh, here, change here, and then here if you wanted to.
Thank you.
Please.
All right. Thank you, Natalia, for that introduction. So, hi, everyone. Like Natalia said, my name is Kassidy Troxell, and I'm a research assistant professor in the Institute of Environment. And I'm going to share with you some of the research that we're doing. So we're all here today because Biscayne Bay is one of South Florida's most treasured and valued ecosystems, yet it remains under pressure from the urbanized area around it. At Florida International University's Institute of Environment, we've been working on ways to understand what's getting into the bay, what it's doing once it gets into the bay, and where it's going. So I'm going to share with you 3 parts of that work here today. Okay. So first, we're going to talk about nutrients. Using 25 years of monitoring data from Miami-Dade County's Department of Environmental Resource Management, along with FIU data sets, we've identified consistent nitrogen and phosphorus hotspots exceeding water quality criteria throughout the watershed. Much of that is linked to the canals and tributaries, which are bringing those higher nutrients, such as the Little River, Biscayne Canal, and the Miami River. The good news is, though, that looking at our data set from the 2000s to the 2025, we've seen that nutrient fluxes have declined as of 2025, though those hot spots in the watershed still remained and need to be resolved. Oh, sorry. I want to go back. How do you go back? No. Okay, sorry. There we go. Okay. So to better understand nutrient transport, you were using a model called Riverflow 2D. It's a model developed and validated by Dr. Henry Bersenio and his team to stimulate the water movement, residence time, and things like phosphorus plume transport. Residence time is especially critical and important for us to understand because we need to really understand what the effects of stagnant water that's coming from the canals and what happens when it sits over vulnerable parts of the bay. And this model is also helping us investigate the conditions that contribute to hypoxia events or low oxygen, and really understand what is happening to the bay when we have those fish kills and massive fish kill die-offs. Okay, so now we know where the nutrients are mostly coming from inside the watershed, but we really want to start understanding where those nutrients are coming from. So we can actually use things like what we call chemical tracers. So these are compounds that are human-made. They're unique to specific pollution sources in the environment. For example, sucralose, caffeine, carbamazepine, a common pharmaceutical. These compounds enter the environment through wastewater. They're present in our wastewater, and when we actually detect them into the environment, we can see where wastewater is infiltrating our canals and tributaries. And we've already identified some specific locations where that's happening, but through a collaboration with Miami-Dade County DERM, University of Miami, and NOAA, we're taking this the next step further. We're actually doing 61 sites throughout the watershed and bay to start really digging into where these nutrient hotspots are and all the different ways we can use to track those sources. So not just using chemical tracers, we're also looking at isotopes as well as eDNA. So we're really just trying to dig into, okay, what is the sources so that way we can actually mitigate them. And last but not least, I want to talk about microplastic and some of the recent work that was published for microplastics in Biscayne Bay. So from our very own Marcellinus Agudo, we detected microplastics at every single site we sampled throughout the bay. It was about 7 particles per meters cubed, and concentrations were highest near the canals and tributaries. And we also found that at low tide, microplastic particle amount was 2 times what it was at high tide, giving us more and more information linking that most of the source of these microplastics is also coming from the tributaries and canals. If you scale up that abundance of the microplastics to the actual volume of Biscayne Bay, it totals over an estimated 18 billion particles in Biscayne Bay, and that's equivalent to over 2 entire football fields. And the most common polymer types that we seen were polyethylene and polypropylene, and those are commonly found in bottle caps as well as grocery bags. And not only did we didn't stop just looking at what plastics were there, what types, but we're also starting to look at the degradation. So these plastics aren't just disappearing, they're fragmenting into smaller and smaller particles. And we did some studies where we put some plastics under Biscayne Bay conditions, and we saw biofouling and surface degradation after 3 months. And then we took these same plastics into the lab and did advanced acceleration with about 4 months of Miami sun directly exposed to them. And there was major fractures and fragments. So these things aren't just disappearing, they're just getting smaller and smaller. And what we're doing in the lab is optimizing our techniques so we can actually track these plastics through their degradation processes and continue to quantify them. Okay. So where are we now? So we began with fragmented data sets. We've now compiled a lot of different nutrient data sets from many different entities. We had no microplastic baseline. We had limited canal source tracking. And today now we have over 25 years of nutrient trends, a working hydrodynamic model, chemical fingerprints of contamination sources, and a baseline assessment of microplastics in Biscayne Bay. And to me, the path forward is pretty clear. We need to continue monitoring in the canals and tributaries, expand the source tracking, and use science-driven management focused not only on the bay, but also on the canals and the watershed feeding it. And the recovery, it really does start upstream. So, thank you.
Thank you, Kassidy. So the next one is Dr. Rolando Santos. He's an assistant professor in the Department of Biological Science at FIU. He's going to be focused on halophila species and also some updates from his lab, the Seascape Ecology Lab at FIU. And his work also underscored the importance of integrating habitat level data into regional assessments.
Thank you. Good morning.
Let's see.
Let's see. Pointer.
And let's see.
What's the clicker? Oh, there you go.
Yeah.
Perfect. So just a little introduction into our lab and our research focus, especially our research focus on Biscayne Bay. So let me find the pointer here. So my lab, as part of the Institute of Environment at FIU, also in collaboration with the Coastal Fishery Research Lab that is led by Dr. Jennifer Rehage, we use this integrated approach where, oh, there you go, thank you.
You too. You can take that out.
Perfect, all right.
Okay.
Yeah, so we try to understand the spatial patterns of habitats, especially how those spatial patterns change through time and space. And then what is those changes, what is the implication of those changes in terms of the ecological processes of the bay, and how those ecological processes then link to certain ecosystem services, especially in the context of fisheries ecology on the bay. And then we take this integrated approach to understand how these different links, right, affects the ecosystem functioning, especially the functioning of the bay. Right, so now I'm going to have 3 slides presenting, look at a snippet of our research on the bay using this integrated approach. So this is a project that was funded by EPA, and we integrated remote sensing, seagrass mapping, the monitoring data from DERM, and what we saw in this study is that there's changes on in terms of the high seagrass cover habitats, these very dark greens in here, right, that I'm also highlighting with these gold box, have changed, right, from 2016 to 2025. And the changes, either positive, right, gain of those habitats or loss of that type of dense seagrass habitat, it is spatially explicit, meaning that there's unique areas that have gain, right, but other that have also lost seagrass habitat. So again, the seagrass dynamics in Biscayne Bay is complex and dynamics, and the culprits of these changes can be unique across space. Another thing that we based on our analysis is that we identify 7 seagrass seascape states, meaning a unique seascape habitat types, right? And through our project, we saw that these 7 seagrass states tend to be overall stable. However, we're seeing something to maybe still monitoring is that this particular state in here, we have seen what we classify the bare, continuous bare, those areas that tend to have low seagrass cover and dominated by low microalgae cover. There have been an increase on the prevalence of this type of habitat through the bay. And this, how I go back? There you go. So this project was, there you go. So this project was mainly led by our student, Mariana Coppola, and also Dr. Ryan James. All right, so we take these habitat changes, right, and we integrate it with stable isotope analysis to understand what was the implication of these changes in terms of like the food production to different fish species. And what we learned based on this integrated approach that it was led by my colleague, Dr. Ryan James, is that we identify areas that have lost food resources, right, in terms for these major fish species that we see through the bay, that they're either important for fisheries, but also important as food for other species that we care, like, for example, dolphins and other fishery species. Right, so what we're illustrating here, right, the green points, right, illustrating areas that gain some of that potential food production, right, that can fuel these populations, versus the purple areas, right, are showing that areas that lost potential food that can help the production of these fish species. And if you squint your eyes a little bit, there's a little more purple than green, showing that we may be losing potential food production for these four major species. All right, so my last slide on some of our effort. This is a collaboration with Dr. Justin Campbell's lab. So his lab identified the first instance of Alophila stipulacea. This is a non-native seagrass that have been crawling through the Caribbean. And so we partner with FWC to understand what is the distribution and what is the growth dynamics of this particular species here in Biscayne Bay. So what we've seen, we did like a quick, rapid assessment across the bay, and identified that, right, that there's a high concentration of the presence in the north part of the bay, but at least for this, at least with this quick assessment, we didn't identify it in other areas of the bay, which maybe is good news. But if you have more questions about these, they have some of the posters around, both Mariana and Dr. Justin Campbell. Also, we use drones to try to understand what is the past growth of these species, and we identify this is the change on growth of these 3 time points that we use drone imagery to map the patches, and we've seen a very fast growth, especially during the summer month of our sampling. And I think Justin have a little more details on this growth in his poster. All right, thank you.
Thank you, Rolando. So next, we'll be hearing from Dr. Catherine Macdonald. which is associate professor in Department of Environmental Science and Policy, and also the director of the Shark Research and Conservation Program at the University of Miami. Yeah, it might be easier. She will be sharing some updates from shark nursery research and the ISRA application, and offering some perspective on how conservation designation can support long-term protection. Sorry.
Thanks. Got it.
Hi, everyone. So basically, I start every talk like this by saying something like, yes, there are sharks in Biscayne Bay, and yes, Biscayne Bay is in fact important to sharks. Probably this audience knows some of that already, but it feels really important to keep reiterating, particularly because I think Biscayne Bay's public reputation is as this profoundly damaged, somewhat lifeless body of water. And in fact, it's super important for a lot of species of sharks. So work at has shown that Biscayne Bay is a nursery, gestation, mating, or pupping habitat for at least 10 different shark species. That means that female sharks are actually finding their partners there, giving birth there, or spending substantial time there during the gestation and development of their pups. Many of the essential ecological functions of the bay are still present, at least for these species, and that means that it's not just a matter of restoring, but also protecting what is still there and still important and still ecologically functional. What we're looking at here is a large female lemon shark being ultrasounded, and that ultrasound showing her internal pups. Lemon sharks are still being born in the mangroves of central and southern Biscayne Bay. So this year, we contributed to a global effort to identify important shark and ray habitats by the IUCN, the International Union for the Conservation of Nature. It's a global effort to identify unusually high-value habitats for sharks and rays, and this is the year that they're assessing North America and the Caribbean for the first time. Biscayne Bay and its immediate surrounds likely meet ISRA criteria for at least 11 species of shark and ray. That means that the bay represents a uniquely important habitat for at least 11 of the shark species that are present there that are also threatened under IUCN threat criteria. Of the sharks that we work with here, 80% are globally threatened with extinction. We have another 12 shark and ray species that are present in Biscayne Bay's waters. And if it goes through, our proposal would create one of the smallest ISRAs globally that meets diversity criteria. So what that means is that Biscayne Bay isn't just important to sharks and rays on a regional scale, but that globally it compares favorably with habitats that are substantially less human altered. I think it's really important that we talk about Biscayne Bay not only as under threat, but also as really important still and capable of being protected in time to make a difference for a lot of these species. What we're looking at here on the right is a big pregnant tiger shark, and below it, satellite track derived habitat use estimates for 2 pregnant female blacktips. And although it's not as dark on this screen as I hoped, if it was, you'd be able to see that both of them spend almost the entire period of their gestation inside Biscayne Bay. At SRC, we work to monitor shark abundance, their reproductive activity, juvenile habitat use, and feeding ecology. If you were here 2 years ago, you would have heard my amazing PhD student who just finished talk about juvenile great hammerheads and their dependence on bay resources for their first 2 years of life. Biscayne Bay is the only established nursery habitat for great hammerheads on the east coast of the United States, although we've got a few other suspects. And the effects of climate change and warming on nurseries, juvenile health, and shark parasite loads. There's still a lot that we can do to generate better outcomes for both the bay and the sharks in it, but I want to close with a little bit of a story. So at the beginning of this year, I got a phone call on a Sunday night that a bull shark uterus had just washed up on a beach. And sharks are really famous for storing a lot of their energy in the lobes of their liver, right? They don't have a lot of fat subcutaneously or in the muscle. It's almost all stored in the liver. So a fisherman had caught this female bull shark, gutted her, dumped the contents of her guts into the ocean, and the big lobes of her liver had actually carried those uteri to shore. So somebody called us, and it's literally the only time I've been the craziest driver on the road in Miami in my entire life. But I was like, if I get pulled over, I'm just going to tell them it's a shark emergency. Yeah. Shark doctors don't get to do that a lot. So I got there, and this uterus was very actively moving. And we were actually able to release 5 pups alive from this uterus. And Mom's success energetically in Biscayne Bay, that big liver that she had to help those pups' development, is what made it possible for us to get there and let them go. But the thing that I want to note is that, like, people called, right? Somebody said, we should do something here. Every year I get folks in Brickell sending me videos of pregnant bull sharks heading up the Miami River. They pup in canals. They're up the Coral Gables waterway. We've got evidence that critically endangered species like the small-toothed sawfish are using those habitats. And so for every person who doesn't care, who doesn't think, who doesn't act, there are people who are. And I know that being a person who's acting can feel like it's not enough. But it was enough for those 5 bull sharks. They're out there, they're swimming around. So I think the thing that I would say is that the science matters, the science contributes, but that we also have to keep in mind that the actions that we take can matter, even as some of these problems are deeply intractable. And the other thing that I would like to note here is that while I agree with a lot of what the previous panel said about the importance of being able to talk about numbers when you talk to policymakers, I also think that if somebody asks you what I talked about, you're more likely to remember those bull sharks than to remember that 80% of the shark and ray species that we study are threatened with extinction. And so don't underestimate the importance of that other part of acting from care and love that is supported by the science. Thanks.
Great talk. Thank you so much, Catherine. So following that, now shifting a little bit from academia, we're going to have Griffin and Alexander, the Biscayne Bay Aquatic Preserve Manager, FDEP, who is going to be then providing an overview of the preserve and going to share some insight on phosphorus and nitrogen dynamics in seagrass blades and offering actually a bigger picture, you know, where they are in the context of managing the bay in the public trust here. Okay. Thank you.
Yeah. Let's see if this works. Cool. Wow, that was extremely hard to follow up, Catherine. As one of the most incredible, probably, professors at UM, not surprising whatsoever. Exactly what you teach in your classes. So telling the story, and she's right, the story's gonna, stick with us so much longer, and definitely it's gonna stick with me. So hopefully I can get close to what you just did. But my name is Griffin Alexander. I manage Biscayne Bay Aquatic Preserve for the state of Florida for the Department of Environmental Protection. That's the laser. Cool. For a quick map, one of our biggest challenges is people don't know that there's an aquatic preserve. People don't know what an aquatic preserve is. Half the people don't know Biscayne Bay is protected at all. We're lucky if they know about the national park. So here's a map of Biscayne Bay Aquatic Preserve. We manage roughly 64,000 acres in one of the, if not the most populated county in Florida. Roughly 3 million people live around the aquatic preserve, around this gorgeous, beautiful piece of water body that we have. I can't have this talk, I can't talk about anything today without acknowledging my amazing staff and my team that we have. I have 5 people, and together we manage this area. Luckily, 2 of them are here with me today, so please come and see them. We do a lot of work out there, and I'm gonna just touch on a very small amount of the work that we do while we're out there protecting it. So first off, water quality. Probably the big thing in everybody's heads is, what's the water quality looking like? You're gonna hear talks about us maybe being impaired today. Perhaps you might have heard about the RAP. the state putting TMDLs on us or something like that. So here's a small snippet of what our water quality has looked like from our programs for the last 5 years. So we have 2 really robust water quality monitoring programs. This is from our 27 sites of bottle grabs. So we'll go out monthly, we grab bottles of water, and then we send them to our NELAC certified lab in Tallahassee to send us different information. 40 different parameters we get, and we're just gonna touch on nitrogen and phosphorus today. The other side of our water quality monitoring program is we have 7 data sondes out in the water continuously monitoring, recording data, and sending us back water quality parameters every 15 minutes. With that, like I said, we have an extremely robust idea of what's going on in the bay. Literally every 15 minutes we can see it live. So that's really incredible. So first off, on nitrogen. Nitrogen and phosphorus, the big things we hear about every day almost. We are seeing the slightest decrease in trend in the amount of nitrogen in our bay, which is really good. Again, we don't want to say that like this bay is dead. It's not dead. We have a decrease in nitrogen. We have beautiful seagrass. We have bull sharks living in here. So things are looking nice. Over on the phosphorus graph, we also have the tiniest decrease of phosphorus. And if you guys can see that tiny little dotted red line there, that is actually our impairment limit. So from the latest data that we have, and this is literally a graph that we made right before we came in with some of our most fresh data, we are just below the impairment limit, but don't get too excited. We set impairment limits over 3-year monitoring periods, over a 7-year time frame, but our latest data does show that across the whole bay, in the basins, that we are looking at a positive trend in this nitrogen and phosphorus ratios. Now, one thing, drought is not a long-term management solution. We are in a drought that is helping reduce a lot of the pollutants entering the bay through the rivers and the canals. And unfortunately, the rivers and canals don't exactly follow the same graphs that I'm showing you here, but the bay is what we're managing. It's what we care about the most, and we are seeing some improvement, which is really exciting to me at least. Next, we have seagrass coverage. On top of our water quality monitoring, to get a bigger picture of the life and everything that's out there, we monitor seagrass. We have about 35 to 25 sites that we do annually, twice a year, I'm sorry. And we do them predominantly around the rivers that are shown here and out in the bays to really see what the seagrass is looking like. A unique program that we do at the Aquatic Reserve is we send our seagrass samples to FIU to be categorized and monitored for nitrogen and phosphorus ratios within the grasses. So seagrass are really cool. They're like a library. They're a history of data, what the bay has looked like since they've been living and since they've been pulling up the nutrients out of the soils. So in the last slide, I showed you our water quality monitoring, exactly what it looked like on the second we grabbed the bottle that day. These seagrass tell us a story for their entire life, which is really exciting. So on the, I guess you guys is right, you can see a graph of the different nitrogen and phosphorus ratios throughout the bay. Blue being higher nitrogen over phosphorus, and yellow being closer to equal. As you can tell, there's some trends and points of areas that there's higher nitrogen that we can probably, understand why. But overall, we're seeing balance, and that's what the bay needs. The bay needs a balance. It might not be about drastically reducing nitrogen without reducing phosphorus, or drastically reducing phosphorus without reducing the nitrogen. Everything needs a balance, just like we need a balance in our lives. So the seagrass does too, and we're really working to find out what that balance is and how we can best provide for all the species and the organisms that we see in the bay. Because we have a little more time here, I do want to touch on a bit more of a social sciencey program that we have. A lot of times when we talk about water quality, we don't often talk about pollution or garbage or trash that's in the bay. And I know that's one of Dave's favorite talks. So I really want to highlight our Leave No Trace program that we started. So within the Aquatic Reserve, we have about 24 spoil islands. Most of them were dredged from when they dredged that intercoastal waterway. And they're places that people love to recreate. They go out, they have a good time, they enjoy and make memories with their families, and then leave their trash when they go home. So a few years ago, we decided that that's not what we want Miami to look like. We don't want these piles of trashes on this island. We don't want people showing up here with their kids and their kids learning that's what they should do. So we went into a really great partnership with the city of Miami and the Leave No Trace program, Miami-Dade County. We got together and said, what can we do? The city of Miami spearheaded this program on their islands. Pace Picnic Island, probably one of the most notorious party islands down south, kind of right across from the Frost Center. It was crazy how much trash was there. The city said, we're going to stop this. They removed a lot of the trash cans from the islands, removing the message of, we want you to leave your trash here, turning it to a message of, bring your trash home with you. At first, it didn't work great. People didn't get the idea. They still left a lot of trash. But then the city made an incredible step and shut the islands to everyone over, I believe, Memorial Day weekend, saying, like, we're taking this seriously. They opened the islands back. It's been about 2 years, and the island looks really, really good every time we go out there. We've moved that program throughout the whole bay. To my knowledge, except maybe 1 or 2 islands, there's no more trash cans left on these islands anymore. Everybody is asked to bring their trash home with them. We started this program pretty recently, again, at Sandspur Island. Many of you might know it as Raccoon Island. It's the large island up by Oleta River State Park. And we've seen similar, really incredible effects there. So water quality and enjoying the bay is a really big picture. And that path to recovery is more than just, science or data tables. It's really teaching the people to enjoy and love the bay for whatever reason they're out there doing, whether it's enjoying the day with their family or fishing, or even seeing birds or enjoying the seagrasses. I mean, a lot of people go out to snorkel or enjoy a day on the reefs. Come into the bay, enjoy a day, snorkel through the seagrass beds. You'll be really amazed about all the stuff you see. When you're here today, please stop by our sponge restoration poster. We also did a novel new sponge restoration pilot project in the bay that we just finished up. So I don't want to take too much time today to talk about that, but please stop by our poster. We're really excited about the progress we made with that as well.
Thank you, Sam.
Thank you.
Thank you, Griffin. So for last panelist, we're going to have Laura Eldredge from DERM, who will be providing an overview of the trap removing efforts, Biscayne Bay report card, and extensive monitoring restoration work being conducted by the DERM team. Their long-term data sets and management actions are actually form the backbone of many of the decisions and recovery strategies shaping the Bay today.
Engagement. This is the face me just go. Okay.
Ooh, good morning, everyone. How's that traffic? I'm Laura Eldredge. I'm the Restoration Enhancement Section Manager at your Miami-Dade DERM. I'm lucky to manage a team of 22 staff who work on restoring, enhancing, and monitoring all of our habitats and species living in, on, over, under, and along the coastal edge. Today we're going to speed around a little bit of scenes from the field. But first, I want to talk about how we actually just released our 2026 report card. Feel free to scan the QR code. So this is a graphic representation of the health of Biscayne Bay that we produce every year since 2019, concerning both water quality and habitat quality, utilizing indicators. We then measure that against, or actually overlay that against 9 of our nutrient regions, and they're presenting that as a stoplight assessment. So that's that yellow, red, and green score. And this is representing the information from the 2025 monitoring data from my team. So scan the QR code and go check it out. Biscayne Bay's benthic habitat monitoring program has been in existence since the 1980s and is a major contributor to the report card and to the reasonable assurance plan, which you'll hear later from Pamela. Staff sample around 450 sites annually to map seagrass and benthic resources. Staff have developed rapid response protocols to survey blooming macroalgae species like Anadiamini or Caulerpa, you've heard of them. Last year, DERM collaborated with FIU for Caulerpa morphological and genetic identification information. So please attend Galia's presentation today to learn more about how our algal cover is actually changing.
Yeah.
This is a picture of the invasive Halophila stipulaceae, which you heard Rolando talk about earlier. We actually found this during an artificial reef survey at Reefline. We've actually gone back and we found that it's now gone, we think likely due to wave action. Our spatially rigorous database, as you've heard about, is helping to find its presence and sometimes more importantly, its absence throughout Biscayne Bay. And we're working in a highly collaborative process, as you heard Rolando mention, with DEP, FIU, and FWC in order to track it, along with other surveying partners. This is the ubiquitous cute photo that I had to throw in. We do actually conduct aerial surveys of manatees. These are population surveys as an estimate of abundance. We also collect opportunistic sightings from the public, so you can submit those to us. And we get some little cute swim-bys from time to time. Our marine debris removal team conducts a never-ending effort. We've had 40 marine debris cleanups in 2025, engaging over 60 individuals who are performing court-mandated community service due to their environmental offenses. This was 12 tons of marine debris removed in just 2025 alone. We've removed 68 derelict vessels solely by DERM since the last summit. About 600 derelict fishing traps were removed since the last summit as well, and many also from our offshore waters. The team also works heavily on illegally floating docks, moorings, and other bulky marine debris items that are also targeted, with several tons having been removed. Staff are also regularly cleaning up golf balls. Yes, golf balls from Crandon Golf Course. In the last 2 years alone, we have removed 10,000 of them. Our habitat restoration team works to restore the coastal edge. This osprey nesting platform installed by DERM several decades ago on Monument Island is really important. It's also struck by lightning recently, so we're working to reinstall 11 platforms along 10 islands in North Bay this year. So this is really important because this mated pair was the first in DERM's historical record in North Biscayne Bay for having a successful nesting, and they've returned every year since. We also have this osprey, which is perched with its catch on a snag from an Australian pine in the Bill Sadowski Critical Wildlife Area, where we are permitted by the state in order to conduct restoration efforts, remove invasives, and plant native species. We plant natives and remove invasives throughout a multitude of the spoil islands across Biscayne Bay. This week, actually, today, I have staff out at Matheson Hammock, where we are planting 15,000 mangroves. Yes, 15,000. This is a 6-acre site where we have conducted restoration efforts by clearing invasives and solid waste, and this is part of just one phase of a 5-phase project where we're going to be restoring 90 acres at Matheson Hammock. DERM also maintains a robust water quality monitoring program since 1979. We had a 30% increase a couple years ago, so now we're conducting monthly assessments across 137 sites throughout the county. Edward will be presenting later, and Charles actually has a poster, so go check those out if you want to hear about our recent data information. Recently, DERM has also facilitated investigations, multiples of them with FIU, not only looking at spatiotemporal dynamics of phytoplankton, but also PFAS within our watershed. So we'll have more information on that in coming years. We also have a new initiative where we're refining the collection of PAR data. So this is thinking about the light availability and light absorption. So DERM will be the first government agency to upload the light data into the state's repository to be utilized for assessments. We have mangrove research as well. So our mangrove research projects include field observations with drone for LiDAR surveys, where we're estimating the models of above ground vegetative biomass. We also have below ground data, and we're assessing that via collected soil cores, as you can see below in the picture. We have a multi-agency academic research initiative with FIU and Biscayne National Park, where we're focusing on mangrove soil development, carbon dynamics, and the effects of urbanization and management interventions. We also have a reef program where we conduct reef preservation work. And so this includes things like installing mooring buoys and maintaining them. We did an expansion a couple years ago. We now have 54 across 14 sites, and we utilize these to decrease anchor damage, or what we call multiple cuts, onto our reefs. Marine debris was removed across 47 of our dives in 47% of our dives in 2025. So for fishing relating debris like monofilament and hooks, that was 24% of our dives. For general trash, cans, plastic bags, 18%. For our line debris, 15%, and trap debris, 10%. We also assisted DEP's coral program. We do damage assessments. So these are assessments that have to be conducted in order to collect evidence for the Coral Reef Preservation Act. And this is where like large vessels can actually throw their anchor and damage the coral or sponges. It can also be caused by unknown large buoys or cable impacts. We've completed annual monitoring at 106 of our individual artificial reef sites. These are offshore and inshore in Biscayne Bay. We also conduct fisheries assessments, like for Goliath grouper presence, which is a part of a larger statewide effort. So I've got a lot of my staff here today. Please reach out to us and get to know your DERM team. Thanks.
Thank you, Laura, so much. Thank you, actually, all of you guys from the panel. Such a comprehensive and interdisciplinary, views of the bay, right? Chemistry, we have conservation, ecology, management. So we have a couple of questions for the panel. I think I'm gonna you answer what you think will be I should have changed that. Okay, sorry, thank you. If you have any for those two questions. So in your perspective, what do you see as the most important research gaps that we're still facing, trying to understand the drivers and impacts in the bay? And then if you try to look ahead, so I know that it's two questions, two parts, so and then what do you see as the most critical steps or the best pathway forward in trying to support the bay recovery? I'm going to start with you, Laura, since you're here next to me, and then we go after that.
It's not like, let's forget it. Turn it on.
Great question. In terms of research gaps, right now we're working heavily to know a lot more about the sources of our water quality impacts. That's something that DERM's putting a lot of efforts towards in our partnership with Kassidy, actually. So that's a big thing for us. Where's the particular outfalls of concern? Another thing we're really interested from the water quality perspective is flow information and residence time, and how impactful that might be for plankton blooms, harmful algal blooms. We obviously need more information on where our invasives are, like the Stipulacea effort. We're going to be following that more and more as we continue to collaboratively work and cross-compare all of our data. So we're hoping to get more information and have a little bit more demos. I've already been initiatives actually removing the invasive by hand. So FWC and FIU have led that effort. So we're hopeful that is more successive and that can be more expanded. But we rely, like Catherine was saying, on the public actually telling us when they see it. So that's critical to that effort. A big thing for us is currently, I'll do a little bit of a plug. 5 days ago, we initiated the next season of the fertilizer ordinance. So we are currently in a fertilizer ban right now. So please make sure that you're thinking of that when you're picking up things for your lawn, for your neighbors, maybe for your HOA. So we want to make sure that everyone is just well aware of that ordinance, the ban itself, and how that could potentially affect things. We've seen some promising water quality data, like what Griffin has said, that ordinance has been around for a few years now. So we're hopeful that will become more impactful over time as people become more aware of it.
Thank you. Rolando?
Yes, in terms of data gaps, and yes, one particular aspect is the fisheries and fish populations, and there's several monitorings that are happening, but I feel that they're a little bit disconnected. But there have to be a concerted effort to understand how these changes that Laura is describing, such as nutrients, blooms, chemicals, microplastic, translate to fish populations and production of there you go, thank you, sorry. of species that we're interested, especially within the fisheries context. That's one area that I think we should like look more into, and so another aspect is not only water quality but also there's been some mention on the need to monitor groundwater, especially in the near shore, because there's some areas that potentially are sources of nutrients through groundwater fluxes. So, anyways, that's.
Thank you, Rolando. Kassidy?
Okay, so just to reiterate a little bit what Laura said, we really need to, the biggest data gap to me is going up into the tributaries, figuring out what, where, and also when. So what are the sources? Are they different in the wet and dry season? And specifically where? Are they major outfalls, like major stormwater outfalls? Is there areas of, different old compromised infrastructure? And there's a lot that we need to investigate in the canals and tributaries, but also a really critical data gap is flow. We need flow. We need flow in the rivers, tributaries to get more accurate estimates on loadings of nutrients from each of them to the bay. And Rollo mentioned groundwater. We are just scratching the surface of groundwater. So we really do need to dig into different areas inland and understand the contribution of that groundwater to the bay as well.
And just, Kassidy, just a follow-up question for you. Like when we talk about chemicals, right? Like, and you talk about for microplastics, and we have like sucralose, and I mean, I have been working on PFAS. Like, in your point of view, like, what are the chemicals you think that might be important to be monitored that are not yet being monitored in the bay?
Yes. So. PFAS, of course, but you guys are digging in the tributaries with that one. There's also things like flame retardants that are really important to understand. Emerging contaminants like 6-PPD and 6-PPDQ that come from our tires. When a car drives down the road, your tire wears off the particles, and that additive 6-PPD actually leaches into the water. In the Pacific Northwest, they have found that concentrations of the 6-PPD, 6-PPDQ have been linked to a lot of the coho salmon deaths. So it's really important that we understand that down here. We have a lot of cars, we have a lot of traffic, so we're going to start exploring and looking at those contaminants. But yeah.
Thank you. Griffin?
Cool. So for data gaps, I think one of the biggest things is recognizing that Biscayne Bay isn't a natural ecosystem that we may have seen, when it was freshwater before we dredged Oliver Canal or something like that. And so we need to have a lot of specific research for this area. So one of the biggest things I would recommend, or I see as a gap, is understanding the specific ratios of nutrients and all kinds of different things, freshwater, pollutants even, that flows into the bay that each of our specific species need. Because I have a hunch that our seagrass don't need the same things that seagrass in a pristine Florida bay might need or have adapted to. So one of those big things would be understanding those ratios to our balanced bay and being able to provide that. Because we kind of provide most of the fresh water to the bay through the canals. So we're able to, I don't want to say change, but work on the flows and the ratios that we do provide to balance our bay in the best way possible. And the second gap, I would say, is more of a human gap, an education gap. We need to get out to the people and educate them on why they should be doing these great things for the bay, why they shouldn't be leaving their trash, where they can put their trash better, what they can do to be the best for the bay, and why they should be doing that. And then what was the second part of your question?
Oh, so what are the critical steps or pathway for recovery of the bay?
Yeah, absolutely. Again, that I think one of the most critical steps is education, teaching everybody why they should be doing these things, to love the bay, to protect the bay. And then, I don't want to say more monitoring, but more targeted monitoring. Areas that we might have physical spatial gaps. monitoring on different populations, maybe indicator species like sharks. We do a lot of work on seagrass and water quality, but what are some of those other species that we need to start monitoring to understand the ecosystem as a whole?
Yeah.
Thank you. Yeah, I agree with that. Catherine?
So I'm going to aggressively speak against my own interest here and say that while if anybody's interested in funding shark research, come talk to me, please. I think actually the biggest data gap for the Bay is in social science. I think everything that somebody here has mentioned would be great for us to know more about, but I think how people are using the Bay, why they're using the Bay that way, and what levers exist to adjust the ways that they use the Bay is the biggest question. I'm very fortunate to collaborate with social scientists, including Dr. Julia Wester at UM, and she definitely I've heard her say to students 1,000 times, conservation problems are people problems. And she's right. The problem for sharks isn't shark related, it's people related. The problem for water quality isn't water related, it's people related. And so what are the things that we can do? What are the acts that we can take? Because I think the task force panel did a nice job of pointing out that the problem isn't that we don't know what the problems are, or even at least to some degree understand their drivers. It's that we're struggling to act on that information. So I think that bringing more social scientists in and treating them as important parts of the science conversation, as opposed to just as like advisors for advocacy, is the big next step for us.
Yeah.
Thank you. Thank you so much. We're good. I mean, I don't have any more questions, but I was going to say, like, if anybody has any final thoughts to share, that will be the time. No? Okay, good then.
I got one.
Oh, you haven't?
Oh, yeah. So if everybody wants to join us on our path to recovery, the governor a few years ago set up the Biscayne Bay Commission for the state. They meet quarterly to oversee Biscayne Bay management, the Biscayne Bay grants that we get quarterly or annually, $20 million a year from the Protecting Florida Together. So come join us at our next Biscayne Bay Commission meeting on May 27th. It's going to be nice and easy. It's going to be virtual for you guys. So join us, follow us on our path to recovery there.
Yeah, and I mean, one of the things I was going to say, like regarding like understanding not only water quality, but I mean, my research group, we have been also looking into the like fish and like exposure to chemicals to fish as well, and oysters, things like that. So I think, yeah, that's something that definitely needs to be more expanded to know more like how probably like wildlife might be exposed to chemicals in the bay. So thank you so much, everybody. And I guess we have a break. Are you going to announce a break? Okay, yeah, sorry.